So hopefully we won't have that looping issue again, but.
Yeah, it won't be an issue now.
Whew. Yikes. Solving things at the last minute. That's good.
That. It's going. Don't ask me. You guys are the ones.
No.
All right. Should be working.
Okay.
I'm going to make you host.
So if that happens again, we know how to solve it. Basically, you got to go in and. Okay, so we're good now. All right, let's see. Let's get started. I assume everybody can hear me. All right, let me get started here. Chase, can you see my screen here?
Yes.
All right, awesome. Everybody, it is my pleasure today to introduce Chase Clark. Chase is going to be defending his dissertation today. So thank you very much for tuning in, and sorry for some of the technical difficulties. So let's get right into it. Chase came to us from, he got his BS degree at Berry College in Georgia, and then he did a little stint in industry at Deerfield Enzymes. And then he joined UIC to do his grad program. Now, these are some of the earlier photos of Chase. I think this is the earliest known poster presentation I see of him, and he was presenting his work at a meeting. And very interestingly, his first project as a student is going to be the last thing that he finishes up today. This awesome work that he's doing with the sponge, and he'll end up telling you about that later. But I thought that was really, really interesting, the fact that he started it, did a ton of things in the middle, published them, and then is kind of returning full circle to the one thing that he originally started with, his sponge work. Okay. So Chase won an award to be trained as a dry suit diver, and I was personally there to see his training. And his very first open water dive was actually in the middle of a fjord in the freezing cold water in Iceland. So he's a very brave student, and he was able to come with us on this project that we had with Dr. Cecilia Omar-Satir from UI, and her students and my students, Dr. Sophia Costa. And so these are just us gallivanting around Iceland, and this is Chase. He was being filmed by a film crew from the London Science Museum, and some of his work was featured as part of that exhibition for a year. And so we were able to do some pretty cool things in Iceland. We were able to dive between the cracks of the continents. This is the Eurasian continental shelf, and that's the North American continental shelf. And this is our entry into that, as we had to jump in. And Chase was able to go and do some pretty awesome things. And he did not just do collections in Iceland. He also went on an expedition with us to the Apostle Islands. And one thing I will say about Chase, and it's very difficult to tell on this video because it looks like, it looks like nothing much is happening, but that's the point. What I want to say about Chase is that you'll see a lot of awesome science today, but he is an amazing dive partner. And for somebody who has had many dive partners over the years and had many problems, Chase always put himself in the right position and the right time. And this video really captures it. He knew exactly what to do, and he allowed me to focus on some of the work underwater as opposed to focusing on him. And for that, I am going to miss. And so he's got many awards. He's won an ASP Student Travel Award. He won a couple awards from UIC. He was awarded a prestigious NIH F31 fellowship. And really, what I think one of the coolest ones was he recently presented at one of the premier conferences in our field, the Gordon Research Seminar, the Gordon Conference in Marine Natural Products. And before that, they have this all student and postdoc run meeting. And Chase was selected to give a talk, which itself is a great honor. But of all the students who were hand selected to give a talk that day, they voted, and he was one of two students to win this, what I call the Student's Choice Award, and re-give his talk in front of the entire conference. And I think that is one of the best awards that he has. And he's also been selected to present at some very prestigious conferences, the ANFAMA, which is basically in my hometown, Cambridge. And then he was selected to present at a very prestigious St. Jude's Graduate Student Symposium, a user international conference. And Chase was a community volunteer as he worked with us in the Near West Side and Garfield Park to teach students different science workshops that we had put together. So in addition to that, he's obviously had a very productive grad student career. He's been incredibly prolific. He's got 4 co-first author publications here. Some are co and some are just him. But like, it really highlights the interdisciplinary nature of his work, where he came in here with a degree in biochemistry, combined it with his computer science knowledge, and then basically combined forces with a lot of like different microbiologists and mass spectrometrists with Laura. Like Laura has been instrumental in a lot of his work. And so he's ended up with this list of publications, and this doesn't even include 2. He's about to submit 2 additional first author publications in addition to all of these. And so what people know him for nationally is he's the IDBack guy, right? He designed this bioinformatics pipeline, and I won't talk about it. He's going to tell you all about it, but just know that IDBack has users in 36 countries right now, and it is the way that I have benefited from it personally is that, you know, these huge drug discovery companies like Novartis, Warp Drive Bio, Denova, they've all invited me to give talks on IDBack, and I've looped Chase in on a lot of these talks too, because he's just so good at obviously the details of it. So just to show you how effective the program has been, it's really proffered a lot of interest from some of the largest companies that you know, that in the field of drug discovery right now. And finally, one thing that he will not talk about today is that in the last month or so, while he was preparing for his dissertation defense, writing his thesis, he was able to design a natural products locator app for our fields. Now, this app has been already integrated into our field's main website, the American Society of Pharmacognosy, and we designed this map just so that students, and particularly students from underrepresented groups, could readily find natural products researchers in their area. So this is an interactive map, and it's an incredible resource that's going to help people get involved with our field. So already he's had a huge impact, and he's not even a postdoc yet. So Chase recently accepted a position in Dr. Jason Kwan's lab, who focuses mostly on metagenomics studies, and he's at the University of Wisconsin-Madison. And now I'm going to turn it over to Chase to tell you about his dissertation research. Let's see. All right, Chase. Should I make you host, or are you good? Yeah. All right. Okay, you are now the host.
Perfect. Thank you, Brian. I don't think the YouTube stream started, Brian, so everybody here gets the selective part. Not anything on your, Brian, you're looking, it's not your fault. It's not going to work. I do have a couple housekeeping things just to let you know. If you go up, if I am on top of my shared screen right now, then you should go into your settings up at the top, hit more, and you want to, I don't even remember what it's called, but side-by-side view. The other thing is, if nothing's going on crazy in your camera, please do turn on your cameras, because it provides me a little bit of feedback. And yeah, so it provides me feedback. Also, the camera is over here, but if I'm looking at the camera, I'm actually just staring at myself in the TV. So I probably won't look there the whole time. So I just wanted to get that out of the way. All right, Brian, can you see this? Perfect. All right, so today I'm going to talk to you about IDBack, which I spent a lot of time on over the past few years at the University of Illinois at Chicago. And that's a proteomic and metabolomic platform for microbial drug discovery. Like Brian said, we have published a few articles on this. the first article was in PNAS describing the IDVAC method. We went on to publish in the Journal of Natural Products on how to use that for minimizing drug discovery libraries. We then made a Journal of Visualized Experiments article, which has a video and written protocol. And most recently, we published an editorial in M Systems. But before I get into all of that, I just wanted to quickly go over the whole presentation, hopefully in 2 minutes. And I'm going to start off with saying that antibiotic resistance is a growing threat. Most clinical antibiotics are actually chemicals that are produced by bacteria. And I will refer to these either as specialized metabolites in the presentation or as natural products. Those two things are interchangeable. And we've been sourcing these specialized metabolites for around 80 years. And that's significant because it means that it's becoming harder and harder not to waste time rediscovering ubiquitous specialized metabolites, ones that are present almost everywhere. And so finding new antibiotics is going to require differentiating bacterial isolates early and with fast, low cost, and easy methods. These methods should inform future bacterial isolation as well. Right, so today I'm going to talk briefly about my motivation for creating the IDVAC pipeline, give a brief introduction to IDVAC, talk about how we are applying that to increase the efficiency in our microbial drug discovery libraries, and then go into what Brian said, talking about freshwater sponges and using IDVAC to characterize the microbial populations present in those. So last year, the CDC updated their threat report in the United States, where they estimated that every year there's about 3 million antibiotic-resistant infections. And this results in the US only with around 36,000 deaths. 1 second. And about 36,000 deaths. And the updated list is seen over here on the right. I'm not going to spend any time on that, but know that this is an increasing problem, also because our drug pipeline has become a little bit stale. So if we look at the different classes of antibiotics, many of you will probably recognize many of the names on this list. But if I draw your attention to the center of this slide, the timeline of discovery, you'll notice that in the 1930s until the mid-60s, there was a boom in discovery of new classes of antibiotics. But this really stagnated from the mid-60s until the 1990s, and it still really hasn't recovered. And there's a few reasons for that. Some of those were recently highlighted in these reviews, where they looked back at high-throughput screening campaigns at GSK, AstraZeneca, and Merck, where they had moved to screening mostly synthetic chemicals, synthetic compound libraries, and they were screening across multiple campaigns, millions of compounds. And while they did receive some leads, those were leads in isolated enzyme single target assays. And it ultimately led to 0 new leads with cellular activity in gram-negative bacteria. So if you go back and look at this chart of the different classes, I've put stars next to some of these. And the stars represent classes that are produced by mostly bacteria and fungi. So these are natural products, specialized metabolites. And again, you'll probably recognize a lot of these names, and many of you may not have realized that these are actually mostly bacterial specialized metabolites. So one thing that we can kind of take away from this is that bacteria tend to be better at killing bacteria than we have been. They've had millions and sometimes billions of years to figure out the best way to compete with each other, and we have only been doing this for the past 100 years. Right, so labs like ours that do microbial drug discovery have pipelines that usually look something like this, where you collect samples from the environment, grow bacteria associated with those samples, fractionate the chemicals present in each of the bacteria isolates, screen those fractions for biological activity. For us, it tends to be antibiotic activity, but we also do anti-cancer screens as others. Hits that are active go into lead dereplication, and then we try to identify the compounds responsible for the activity. Our lab is primarily focused on improving the efficiency at these first two stages, and that's where most of this talk is occurring in this process. Right, and so labs have trouble prioritizing which bacteria to study. And just looking at a plate like this, which just comes from some sediment smeared on an agar nutrient plate, we may discard the different orange colonies and only keep one, assuming that they are producing the same specialized metabolites because they look exactly the same. Hopefully in this presentation, you'll come to realize that that is not always the case, but this is the by eye process by which many labs choose to retain isolates for further study. Right, and we don't have just one of these plates when we come back from a collection trip. We have hundreds, if not thousands. So to put this into perspective, this is a relatively new slide, but I wanted to show that this is a phylogenetic tree. Here are Homo sapiens, so humans, and here are mice. So the most recent common ancestor between mice and humans would be at this point right here. Between humans and chickens would be right here, rice, etc. But if we zoom out and look at the entire tree of life, you'll notice this large purple section, and this is all bacteria. So the amount of diversity present within bacteria is incredible, and there's no way that we can discern all of that only by eye. I'm not going to get into this either. This is a recent tree of life by the Banfield lab, but all of the bacteria I just showed you encompasses this. We now know that there's actually another clade or phyla of bacteria that I'm not even going to talk about today because nobody has ever knowingly cultured any of those. So bacteria are really diverse, and I just wanted to get that point across. So we need to do something better than just prioritizing and looking at these using gross morphology by our eyes. And to do that, our lab has turned to MALDI-TOF mass spectrometry, which is really fast, cheap and easy, and it allows us to collect data on every isolatable colony. Real briefly, how MALDI-TOF works is we have bacteria on an agar plate, like you saw, touch a toothpick to a colony, smear that on a MALDI plate, add formic acid, let it dry, add MALDI matrix, let that dry, insert that into the instrument, irradiate the sample. This creates an ion plume. And as the ions travel through the time of flight tube, they separate based on their mass to charge ratio. And what you're left with is a spectrum where the x-axis represents the mass to charge ratio and the y-axis is abundance or intensity. What's even cooler about MALDI-TOF is that we are actually able to collect two different types of data for every colony. We collect protein spectra, which allow us to group bacteria based on who they are, and it allows us to collect small molecule spectra. And these are the region where the specialized metabolites are. So if we have a diversity plate like this that has 11 colonies, we group the isolates using the protein spectra, and this is highly concordant with phylogeny and 16S results. You can see the Rhodococcus colonies, the genus, all grouped together, Paenibacillus groups together. For the small molecule data, we use these metabolite association networks where the large nodes represent bacterial isolates and the smaller nodes represent individual M/Z peaks or mass features. And this allows us to quickly see gross differences between isolates like 10 and 7 being very different in their specialized metabolites than the rest of the isolates, but also, while sharing a lot of unique mass features or specialized metabolites, they have their own unique as well. But we needed to prove this was actually the case, that we were measuring specialized metabolites. So we took 2 known isolates or strains, Bacillus subtilis 3610, which is able to produce a series of molecules and analogs of plipostatins and surfactants, and Bacillus subtilis PY79, which has a single point mutation that prevents it from producing these suites of molecules. And if we zoom in to just the raw small molecule spectrum, we see Bacillus subtilis 3610 in the positive y-axis producing these molecules, and in the negative y-axis, PY79 does not. So we are measuring specialized metabolites. And I will point out that we have seen metabolites across the spectrum, including PKS and RPS, among other classes. But we wanted to look at this see if we could discern subspecies level differences. And to do that, we used 8 micromonospora isolates. 7 of these were derived or isolated from the same cubic centimeter of sediment. B031 came from a different part of Lake Michigan. But what we can see with the 16S rRNA phylogenetic groupings is that it's highly concordant with the groupings that we get with MALDI-TOF. And if we look at the metabolite association network for these isolates, we see that while there is some concordance with the groupings here, as with the protein, which I'll talk about a little bit later, we notice that B001 is kind of far off from the rest of the isolates, and that was confirmed through LCMS to be because it cannot produce a series of analogs of desferrioxamine. So this is the most programming slide in the entire talk, I promise. The IDBack Shiny app in our package is available for anybody to use. It's free and open source, and you can find that on my GitHub at chasemc/IDBack. If you're really interested in just getting started with the method, again, our Journal of Visualized Experiments article goes through both the wet lab protocol, but also walks users through each part of the software. Okay, so I made some software, but you may not be convinced that it's totally useful, and that's fair. So through the next 2 vignettes, hopefully you see the usefulness of this tool. And the first is through this expedition that Brian talked about in Iceland, where we used an Icelandic expedition and strains in order to see if we could minimize the taxonomic and natural product redundancy in microbial libraries. And I was going to say that Iceland looks nice and sunny, so you think diving's really pleasant, but just around the corner, things looked like this. So it wasn't always super nice and warm looking. We were often dressed like this. So this was through a collaboration with the University of Iceland. There have been some media reporting on this you can check out later. But the year I'll be talking about is represented by these sampling locations in blue. And we were still creating IDBack at the time, but we knew what we wanted to do, and that was to take MALDI-TOF mass spectrometry data and collect that on all colonies that we could isolate from all of the samples we collected. We would then group colonies by their pseudophylogeny and then view the natural product overlap within each group in order to create a low redundancy library. So like I said, the year I went represented by these blue dots or blue points, and Sofia Costa, a collaborator on this project, isolated, I think it was 1,616 bacterial isolates, which is an insane amount of work, and did triplicate data collection on MALDI. And what we got was this pseudo-phylogenetic tree. She then went in and performed close to 300 identifications via 16S rRNA sequencing, and some of the results of that is shown here. It was a highly diverse set, and we recovered a lot of actinobacteria that are considered understudied. But the manual prioritization step was, I will say it was rough. Selected small groups, here one represented by Bacillus, and then within each of these groups, Brian and Sophia, I did not take part in this, went through just 1 million metabolite association networks and chose isolates that represented that metabolite association network, but tried to remove isolates that would just incorporate redundancy into the library. And so the results of this was, like I said, 1,616 isolates were recovered. After IDBAC prioritization, 300 isolates were retained, and that represented 54 genera. And for people that are familiar with this process, that is just a really high number, especially considering the low number of isolates for that. And that represents an 81% reduction in further work that we'll have to do. So probably much to Sophia's dismay, Brian asked her to go back into the -80 freezer and get all 833 isolates from the previous 3 expeditions and perform that same process all over again. And what came out of that was a 72% reduction in strains that we had already selected manually by eye, morphologically, to try to be less redundant. And it still proved to be highly redundant. So the main takeaway here is that instead of studying close to 2,500 isolates, now we only have to study 534. And that's a dramatic reduction in the amount of work and taxpayer money that we have to spend further along in our pipeline. So during this entire time, I was busy writing IDBAC, but we were all thinking, why isn't this automated? It seems like we could automate it. Why is it not? So the simplest way that you could think of automating this process would be to group the bacteria like before using the protein spectra. You could cut this tree, which would create groups all along this dendrogram or tree. And then within each of these groups, choose isolates that contains the most specialized metabolites up until you capture a certain percentage of the specialized metabolites that are present within this group. And so what this would look like in practice is we would cut the tree, and as we walk down the dendrogram or tree, we're creating groups, and represented within each of these subgroups, you can see the red dots, which represent selected isolates, selected by the algorithm. And that's, again, not selected based on the protein, but selected on the metabolites that are produced and seen in the specialized metabolite spectra. So recently, we've put a lot of work into this. Lynn collected 850 isolates from Vietnam and broke that into 80 different metabolite association networks, and 2 members from the lab manually went through all 80 of those and selected isolates, and then I fed it to the new algorithm in IDBAC. So the humans, or the researchers 1 and 2, the isolates they selected are represented here in blue, and the missed specialized metabolites, or mass features, are in red. And we can see that between researcher 1 and IDBAC, they're perfectly similar. Human 2, or researcher 2, chose a few less here. But if we look at this in total, IDBAC and all researchers selected the majority of specialized metabolites. And IDBAC performed very well and did so with 50 less and about 100 less isolates than the two researchers. So here I have the percent mass features captured within each group. On the y-axis, we asked the two researchers to select at least 75%, or to aim for 75% capture of specialized metabolite peaks. And we can see that the first researcher did quite well. The second one was a little less experienced and a little eager to make sure they chose more of the specialized metabolites. IDBAC is an algorithm, so it did exactly what it was told and tried to optimize for right above 75. So as a brief summary, MALDI-TOF mass spec is rapid, it's low cost, and it's super easy to use for prioritizing bacteria for drug discovery. IDBAC can be used as exploratory software through the Shiny app or as an R package. And that allows nearly complete automation of the prioritization of isolates. And that's based on actual chemical data, not just based on people's eyeballs and morphology. So I'm going to pretty drastically shift gears into another use that we're using, or another way that we're using IDBAC, which is to study the metabolite and taxonomic phylogenetic diversity of the microbiomes that are present within the freshwater sponge, Eunapius fragilis. So this is actually a longer term project. It's been going on for about 5 years. We have 28 citizen scientists, and between them and us, we have collected 93 freshwater sponges from across the Great Lakes, which encompass 3 different sponge genera. Today, I'm going to talk about 2 of those sponges shown here on the right. These are Eunapius fragilis variety minuta, and they were collected about 1.5 kilometers apart from each other. And from these, myself and an undergrad at the time isolated 851 bacteria. We then, the incredible amount of work, this is about half of the plates that we used, is an incredible amount of work, but then we used IDBAC to try to document the diversity present within those isolates. So on the right-hand side, I have a pseudo-phylogenetic dendrogram made from the MALDI-TOF protein spectra. And I'll draw your attention just to a couple things real quick. There were 2 genera, Delphtia and Chryssobacterium, which constituted a large portion of the isolates that were recovered. But we did also recover isolates known for drug discovery and for antibiotic production. Paenibacillus, Streptomyces, Bacillus, Micromonospora, and others that we may not have identified. On the right side, we've plotted different media conditions or isolation conditions that we used. Here we have sample dilution, so the dilution of the sponge sample that was placed onto the agar media, going from no dilution, 1 in 10 dilution, and 1 in 100 dilution. But what is most striking in this graph is the pre-treatment by heat, which selects for endospores, so bacteria that are able to take a spore form that allow them to survive in extreme conditions. So here we notice that when the samples are not heat shocked, we grow a lot of gram-negative, as can be expected, or recover a lot of gram-negative. And for the samples that were heat shocked or heat treated, we do select for spore formers like Micromonospora and Bacillus. Looking at the media type, the trends are a little bit less clear, but I will point out that Micromonospora was recovered mostly from low nutrient agar plates. What's, I think, more interesting to me is if you look at the Delphtia genus, there seems to be some species to subspecies preference for media type. And that is also seen within the Chryssobacterium genus as well. So for those less familiar, here's a taxonomic tree to the left. I'm not going to go into it too much. I just wanted to point out that we were able to recover 4 different phyla of bacteria that are commonly associated with sponges. And in order to identify the bacteria, we used 2 different methods. One was 16S rRNA gene sequencing, but we also complemented that with MALDI-TOF database searching. So searching MALDI-TOF spectra that were collected on bacteria whose identity we know. And to do that, we use just a simple algorithm, cosine similarity, and the results of this worked out pretty well. So if we look at the 16S assignments, the bacillus down here match up perfectly with the assignments with bacillus by MALDI-TOF. And we hadn't sequenced any Micromonospora samples, but it's pretty obvious that this pseudo-phylogenetic group is Micromonospora based on the MALDI database matches. And that also is confirmed morphologically because Micromonospora are very distinct morphologically. So we next had the question of what the variation was between culturable bacterial populations between these 2 sponges. And just to throw the answer out there first, we saw that less than half of species level groupings were shared between the 2 sponges. The simplest way that you can think of that comparison would be by a Venn diagram, where you have isolates or groups that only contained isolates from sponge A, only contained isolates from sponge B, or pseudo-phylogenetic groups that contain isolates from both. So if you're using 16S, you would use something like an operational taxonomic unit. We don't have that here. So the best thing we can do is to cut a dendrogram and create groups. So like this cut would create this group, cut here would create this group. But another problem with MALDI is that we don't really know where to cut the tree. That's not straightforward. And so what I did was cut the tree at intervals of 0.05. And at every interval, see whether the groups contained isolates from one sponge, the other, or both. And what we can see on the right is that at approximately 2 would be a genus level cutoff. And below this, the majority of isolates either come from one sponge or the other, and less than half of the pseudo-phylogenetic groups at the species level contain isolates from both sponges. We also did this for just the raw isolate count as well. It's a little bit less informative, but necessary. To move forward, we also wanted to look at the specialized metabolites and see whether any of the isolation conditions or other factors influence the recovery of those. And so for the metabolite association networks, I broke out 6 pseudo-phylogenetic groups: Bacillus, Chrysobacterium, Delphtia, Micromonospora, Pseudomonas, and Streptomyces, and colored isolates based on some property. And what I discovered was that the largest factor that we saw to influence recovered specialized metabolites was actually phylogeny. So if we look at the pseudo-phylogenetic tree for Pseudomonas, these are all Pseudomonas isolates. They're colored into 5 groups. And if we look at the blue and purple isolates in the pseudo-phylogenetic tree, we see that these also group together in the man. The yellow isolates are clustered both in the man and the tree as well. To be a little bit more quantitative, we calculated this both for the entire data set, but also for these 6 groups. And you can see that some genera have really strong correlation between protein spectrum similarity in the y-axis and small molecule spectrum similarity in the x-axis. On the other hand, some groups do not, such as Delphtia and Chrysobacterium, which brings me to the point that the factors that influence recovered specialized metabolites is phylogeny, but it's a complicated relationship. So for something like Chrysobacterium here, if you look at the green and red isolates represented on the left, you can see that they're all mixed together, but they are quite distant in the phylogenetic tree. So for some groups, some genre, specialized metabolism was found to follow phylogeny, but for some groups, it's not that straightforward. So as a brief summary to that, while some bacterial genera were recovered from both sponge samples, we observed significant variation at the species level. But as a glass half empty, glass half full note, that also means that a little bit less than half of species recovered were shared between sponges. Additionally, phylotype and chemotype were often correlated at the species level. That aligns with previous genetic studies. The easiest method to increase specialized metabolite coverage from bacterial isolation would therefore be to increase phylogenetic diversity at every level, at every taxonomic level. And then lastly, recovering multiple isolates of the same species requires differentiation with higher resolution than what 16S rRNA provides. So if you are using 16S to guide your discovery pipeline and you're throwing out isolates that have perfectly identical 16S, you may be throwing away isolates with different specialized metabolites. And so this is my last slide. This is how I envision this drug discovery process with IDBAC, where you collect samples from the environment, you culture bacteria associated with those samples, you perform MALDI and IDBAC on those isolates, and you use the protein and small molecule spectra in order to create diverse libraries. But additionally, this wraps back into your sample collection strategy, and you use that data to inform your future collections. All right, so I have a lot of people to thank. I'm gonna miss people, I know I am, but here's the most recent lab picture that we have. So our lab has been fantastic. The people in it are amazing. I've listed here current grad students and postdocs. There's a whole slew of postdocs and undergrads. I'll get to them in a minute. Dr. Laura Sanchez, I'm not sucking up because she's on my committee, but she is the MALDI-TOF guru, and she has been instrumental in making this pipeline happen. My funding sources are, like Brian said, the F31 from the NCCIH. IDBAC is also funded on an NIGMS R01. The freshwater sponge research was additionally funded off of a National Geographic grant. The department and especially the Center for Biomolecular Sciences have just created a really rich and engaging program at UIC that has, bar none, been the best. So the American Society of Pharmacognosy, this is weird to maybe thank an organization, but I attended a meeting back in 2014 when I was still working at Deerland Enzymes, which was an ASP meeting, and it's the only reason I heard about UIC. And so without ASP and their meetings, I would never be here standing in front of an empty room other than my wife. I actually saw IDBAC has built off a lot of previous technology and work. One of those is the package MALDI-Quant, which was written by Sebastian Gibb, who I actually saw is in the call. Thank you, Sebastian. If this package hadn't been written, I would never have even endeavored to get into writing R. And so just this being available set my path onto where it went. There's a lot of people that whose software and IDBAC uses or builds on. I'm not going to name names here, but if you want to know who these people are, look at the IDBAC GitHub. Additionally, the RN Pharma conference really has been fantastic for the past couple of years. Especially wanted to mention Phil from our studio, who went out of his way to get me a travel award to attend the first year of RN Pharma. This has been an amazing group of people to get to know and interact with over the past 2 years. Additionally, the Chicago Python user group and our user group, especially the R user group, has given me the opportunity to increase my public speaking and really given me a platform to increase my work. For the RN Pharma conference, again, it's been great. I've been able to meet a lot of people like Olga Vitek, guru of mass spec statistics, Joe Chang for ours, Shiny, and then people like both Max Kuhn and Leon, who are experts in machine learning for pharma and R. So the lab, like I said, is big. I'm sure there's people that are missed here. This is mostly the current members and some past grad students. I'm not going to name names because I'll miss people and get too emotional in case this is still on YouTube. But our fearless leader here, Brian, has been great. I don't think Antonio was on the previous slide. I wanted to point him out because he did a ton of the isolation work for the sponge project that I mentioned. He is now a grad student, so I guess I didn't scare him off too bad while he was an undergrad. And here he is plating samples, or we're plating samples for the sponge work that he is performing. I put this in here. This is actually one of my first days during my rotation at UIC. Michael Maloney was a previous grad student, now a postdoc in Neil Kelleher's lab. Joel was an undergrad at the time, is now a grad student at UW. And I like this picture because it shows exactly what the process was before IDBAC. It was people in a room staring at plates, trying to decide which bacteria to choose for further study. Sophia, like I said, did a lot of the work for the minimizing libraries. This is us being shunned into the library or guest room in Iceland. Brian made us work one day on IDBAC so we could get the publication out. But Iceland is a great experience. I can't speak highly enough about it. It's a beautiful place, and we were able to meet a lot of people who really made the experience just fantastic. I realized the privilege of getting to do scientific expeditions, especially overseas, and it's not lost on me what a unique and amazing opportunity that was. A lot of my pictures show Brian being really serious. That includes when diving and coming out of the water. But he's not. He's a really happy guy most of the time. Here he is coming out of the freezing cold water of Iceland. We've done a few sample collections together, including Iceland and the Apostle Islands. Here he is looking in to the depths of a wastewater treatment facility where there actually are freshwater sponges growing. And he's also just trying to push us all to climb to new heights and showing us that it's okay sometimes to fail. This is us in the Apostle Islands. And again, Brian, thank you. I did want to say I was working at Dearland Enzymes for a few years. I applied to 5 different grad schools. My undergrad grades were like, meh, okay. I only got into one school, and it was UIC, and Brian took a chance that a lot of other people did not. So, Brian, thank you. I got to move on or I'll get emotional, but thank you for taking this chance. The only other person in the room right now, it's really weird giving a seminar to an empty room, but my wife is here, and I want to especially thank her. She agreed to move to Chicago. We're both from really warm places, and I think this was probably her face when I mentioned moving to Chicago. But it worked out, and I think that it's worked out for both of us, and we're a lot happier now. we've been going on some crazy adventures over the past few years, and I hope those continue. My parents have also been super influential and supportive for the past 30 years. And there's no doubt in my mind that I wouldn't be here without them. And I know I missed a ton of people to thank, probably most of the people on this call. There's a lot of people in Tennessee, California that I've missed. But with that, I'm going to say thank you, and I'll take any questions. Brian, I have made you the host. If Brian is around.
Yes, yes, I am around. Yeah, so please, any questions for Chase? Let's, I can unmute you, or you can throw it in the chat also.
Hey, Brian, do you want make me the host again? And I'll just leave your audio on. Actually, no, never mind. I just don't know how we're going to do the slides. I'll try to answer without the slides, and if I need them, we'll go from there.
Shura, unmute Shura. Okay. Oh boy. You know, every instinct tells me not to, but I'm going to do it anyways. Here we go.
Thank you, Brian. I'll try not to occupy too much time here with the general audience. Nice presentation, Chase. And as you can see, the hand picking that Brian used when picking you for the graduate, as a graduate student, sometimes broke. Not probably always, but in this case, definitely did. So I have a question about the value of the protein ID and small molecule ID in picking strains for collection. As you showed in several examples, sometimes the phylogenetic close grouping does not necessarily mean that the metabolites will be also similar. Like with the, I think it was Delphia in case of sponges, where there was no basically correlation. So if there are two phylogenetically closely related species, but with a substantially different metabolite spectrum, would you recommend, or would your software recommend picking them both for the library or not? Say too close.
If I understand correctly, like if you have the phylogenetic group and you make the man and they're like pretty separate within the man.
Yes, correct. Yes.
Yeah, it would choose. So it selects the isolate with the most specialized metabolites. Mm-hmm. It removes all those metabolites from the man that are associated with that one, and then chooses the next one that has the most. So it is a little bit naive to the structure of the man, but yeah, but what we have seen of how it works, then yes.
So then the final criteria is man, essentially, right? So then why do you need phylogeny at all? Why do you need the protein ID? Why not just to do small metabolites and use this as criteria for chemical diversity?
Yeah, that's the question we get the most, I think. And the simplest answer is that the MALDI-TOF that we use isn't a high-res instrument. So you will have overlap of ions. So it just provides us more fidelity, and we don't have to worry as much about overlap of different molecules that have similar masses.
So with a good mass spec, you would be basically foregoing phylogeny. You could go just for the small molecule diversity, right?
Correct. If you had like an FTICR instrument, you could just do the small molecule part. Yeah. The good thing about the one that we use is that it's available in a lot of universities and institutions. So.
Yeah. Thank you.
Chase, we have a question in the chat from Mark Chevret. And then a follow-up by Isabel Cruz. So I'll just read it. Mark says, in the case where phylogeny is not a strong indicator for specialized metabolite content, do similar strains share characteristics, e.g. a niche?
Yeah, I knew Mark was going to do this. So Mark, actually, when I said that previous studies have seen correlation between phylogeny and small molecules, Mark recently wrote a paper on all of that. So he probably knows all of this better than me. I would love to answer that question. We only have data on these 2 sponges. So it's really impossible for us to say with any definitive answer as far as whether things that we're seeing because this one was collected on a rock or this one was collected on wood. We don't have that kind of data yet. But that is definitely a thought moving forward that hopefully Brian or somebody in the lab will look at when they collect their isolates from more sponges.
So then Isabel writes, great talk, Chase. Can you go into more detail on how you used R? I am also wondering if you have captured the workflow in some kind of formal way.
So I can go in a ton of detail. So it builds off of MultiQuant. In the beginning, it was MultiQuant built into KNIME, which was a headache for people to use. I then wrote a Shiny app, which is thousands of lines of code, so there's not enough time to go into the detail of that. But it's really built off of MultiQuant. I've added some things or replaced some things from MultiQuant within that. But now I've refactored it as a package that's also a Shiny app, so it really can be used just as a command line. The biggest thing probably isn't even R. The SQLite database that IDBack creates and relies on is probably the thing I'm probably most proud of out of the thing. As far as capturing the workflow, not really. There's some, for the pre-processing, that process has been captured, but the workflow is pretty open within at least the Shiny app for people to do exploratory analysis. So it's not necessarily a linear workflow in the Shiny app. Hopefully that answered your questions. You're on my committee too, so if it didn't, you can feel free to ask again.
Are there any other questions for Chase? Scott Frensblau asks, is there a risk in assessing specialized metabolites based on a single medium? Ooh, loaded question.
100%. Yeah, 100%. Yeah, that's just how much time and effort expenditure your lab can take on. We do a single media type, but I have slides on it, but it's going to be too hard to show. Where I did grow isolates on 6 different media types, and on one of those media types, and only one, it produces a series of molecules that are specialized metabolites and potentially druggable molecules. So there's always that risk. It's just how, if you think about doing 1,600 isolates on one media type, you add another media type, and now your work has doubled. And so it's just a point of how much your lab can handle.
All right. Yeah, that question's a rabbit hole. Great question. Okay. Any other questions for Chase? Mark Charette, follow-up. If you were to do the same set of approximately 20 strains on 2 different media and clustered them via IDBack, would you expect media to group with media or strains to group with, would you expect media to group with media or strains to group with strains?
I think I get what your question is. And if, by what I just said, in the small molecules, it's going to be a case-by-case basis. Who knows? Sometimes it will, sometimes it won't. For the protein, there's extensive supplemental information on the PNAS paper. And in that, we did show that on different media types, strains do cluster by strains and not media using the protein data. So as far as the protein groupings go, media doesn't seem to be in effect.
All right. Any more questions for Chase? All right, awesome. Well, if not, I think we're going to continue on to the next portion. We'll just ask everybody, we'll ask the committee to stay on this thread, and we'll ask everybody else to sign off. And just Chase, everybody's clapping in their offices and rooms right now, so. I know it doesn't sound like it. All right, let's see. And I wonder if because sometimes there are people who have, I don't know who would do this, but who have left their computer and have gone on to do yard work. I think I have the power to like boot them.
You can boot, but maybe also the group
I
rooms or whatever.
Yeah, I can definitely, I do have the power to like, to remove people. All right, so we've got, here we go. Shura, Isabel, let's see, is Laura here? Yep, Laura's here. Jimmy's here. Is Jimmy in your committee? Wait, who? Laura, Isabel, Shura. Who's the other member? Yeah, Jimmy, you're in this committee, right? Okay, awesome. Jimmy, will I need, oh, you know, actually what I should actually get ready. Will I need,